I talked actually about the scope of neoantigen-specific T cells and T cell receptors. This is actually what we are doing in my research. We have developed in the past a pipeline which can specifically actually validate the existence of neoantigens on the tumor surface. And this is actually a proteogenomic approach where we, on the one hand, perform a deep sequencing analysis of DNA and RNA in the tumor cell to detect the mutations which are expressed, actually genetically expressed. And on the other hand, we are looking by mass spectrometry if potentially mutations are presented as peptide ligands, so associated to MHC class 1 and 2 molecules to the surface, and then can be also detected actually by the T cells. And I think this is a very important step to go a step further, because in case of prediction, you are never sure if those neoantigens which you predict may be in fact presented. And if you do an analysis by mass spectrometry, you can be safe that they are present and can be detected by the T cells. And using this approach, we have previously identified a number of neoantigens in patients with melanoma. And we were also able to validate those neoantigens with one further step, using actually the T cells from the patients. And in one case, and this was a study which I presented actually at the ICLE, we detected different T cells and T cell receptors, which are specifically for 3 neoantigens. And the interesting thing in the story was that 2 neoantigens, or against 2 neoantigens, we have identified a couple of T cell receptors, and these T cell receptors differed in their function. And the function, we saw actually T cell receptors which had a very good function in vitro, very high response with interferon, for example. And we saw T cell receptors which were a little bit less active. And interestingly, those T cell receptors which were a little bit less active in these studies were those which were functionally very, very active in vivo. And what we subsequently analyzed, and I was not able to show all these data, was that these T cells which have a little bit lower activation at the threshold, that these T cell receptors actually are less prone to go into exhaustion. They do express less PD-1, and they are quicker reactivated after restimulation in vitro and in vivo. And the actual characteristics of these T cell receptors are now extremely interesting to us. Why do they behave that way? And can we use that and transfer that maybe to other populations, these characteristics? And therefore, we started now to do an deep sequencing analysis of the T cells themselves from the patient. And we did an single cell RNA sequencing in these patient T cells, which we reactivated with a specific mutated peptide ligands. And what we learned so far is that, in fact, the transcription profiles of these T cells and T cell receptors are different. And there are interesting, actually, target candidates in the signaling of the T cell receptor itself, which actually make them different between the very highly activated T cells, which are certainly great to do the first actually activation step, but also to the other T cell receptors, which may be a little bit in behind, a little bit less activated, but therefore have a longer activation potential and a longer potential to react against the tumor, as they do have a long journey to do to reject the whole malignant diseases.